Welcome, I'm Bérénice Batut, and today we will dive through building and annotating metagenome assembled genomes using Galaxy. Are you ready to dive in? Learning the tutorials hosted on the Galaxy Training Network. So go to training.galaxyproject.org and then you scroll down until you see the microbiome topics here. You click on it and then you follow and you go to the metagenomics sections, that is after metabarcodings, and you find this building and annotating metagenome assembled genomes from short metagenomics paired reads, and you open it. So in these tutorials, we will focus about answering the question, why is it important to perform quality controls and remove contamination from our reads before assembly? What is the purpose of bindings in metagenomics analysis? How does it help reconstructing marks from complex microbiome communities? What are the metrics that we use to assess the quality of marks? How the taxonomic assignments contribute to the analysis of the marks? And what is the significance of functional annotations in metagenomic studies? So you see here the list of objectives, so what you will be able to achieve at the end of the tutorials. And these tutorials will take some time. We will try to make sure so that we provide intermediate steps because some of the computational steps are taking long. So as you know, metagenomics has revolutionized our understanding of the microbiome communities by enabling the studies of the genetic material directly from samples. And one of the most powerful applications of metagenomics is the reconstructions of metagenome assembled genomes, or what we call MAGs, which are the near complete or complete genomes of individual microorganisms that have been recovered from complex microbiome communities. So MAGs provide really important insight into the microbiome diversity, the functions, and the ecology without the needs for laboratory cultivations. And so this tutorial is designed for anyone that is interested in building MAGs from paired short metagenomics reads. And so here we will learn the different steps that are essential from quality controls and read processing to assembly, binding, refinement, and annotations of MAGs. And hopefully by the end of the tutorials, you will be equipped with the knowledge, tools, and workflow to really generate high-quality marks for your own metagenomics datasets. In these tutorials, we will not go step by step by running every tools individually, but we will use Galaxy workflow that were developed within the FairyMarks project, and which is maintained by the Intergalactic Workflow Commission, which is a group of people interested in to build and maintain high-quality Galaxy workflows. And all these workflows are available via the IWC workflow library that I really recommend you to look at to see if you can find other workflow that might be interesting for your study or for your analysis. And all these workflows are then available in workflow registry that are called Workflow Hub and Doc Stores. To run this workflow, we will use data that come from a case study from the honeybee gut microbiome. So we will use the public data from a study where they investigate the response of the honeybee gut microbiota to Nosema serenae, which is a prevalent microsporidian parasite in the bees. And when the response of Nosema under the influence of not probiotics and all neonicotinoid insecticides. So you can find the study that has been published in 2024. We know that the honeybee gut microbiome comprises a relatively simple core community that is dominated by 5 bacterial lineages that include Snodgrassella alvi or Gilliamalea apicola that play essential roles in digestion, immunity, and pathogen defense. So we expect to see them when we do MAGs and rebuilding MAGs on them. But we know that this microbiome is highly sensitive to abiotic stressors such as the pesticide, the pollutants, climate change. but also, for example, the exposure of neonicotinoid has been linked to shifts in the microbiome diversity and is known to increase the susceptibility to opportunist pathogens like Nosema serenae. Usually this research of what is the impact of each stressors is done in isolations, and the synergic effects of, for example, pesticides plus pathogens are usually, the synergetic effects are remainly understudied. And so in these studies of 2024, they infected honeybees with Nosema serenae spores and expose them to a neonicotinoid, or they were treated with probiotic bacteria, which is known or thought to enhance the tolerance to Nosema. And in these studies, they use deep shotgun metagenomics Illumina sequencing to analyze 21 samples and looking at the taxonomic compositions that explored with using MetaPlant4. So we could use this data that has been published as a comparison when we generate the mags there. So that is what we will do at the end of the tutorials. And if you want to redo that, redoing really just the taxonomic profiling, you can follow our dedicated tutorial for that. And in these tutorials, we will focus really on two representative samples from this dataset that is to highlight, to show how to reconstruct mags. So we will use a sample that is called IP2 and the sample IP1, I1. So IP2 has been infected with Nosema spores, so it's N, and is treated with the probiotic, so P. And it's the second replicates. And we know the biosamples and the runs on the SRR numbers for the run. And the second sample is I1, which has been infected only with Nosema spores. And it's the first replicate of these treatments. And we again have the biosample sample run there. And the first things to do is to import the data into Galaxy. So first, please open your favorite Galaxy instance. So for me, I will use the usegalaxy.fr, but you can also use usegalaxy.eu. And the first, or galaxy.org, or use Galaxy, or the Australian one. The first thing you need to do is to create a new history. You create a new history and you give it a name. Here I will call it maths tutorial, and I will annotate with the date. So February 2026, it's when I record these tutorials. So the max tutorials and B microbiome. Let's put that here. Oh, we could also put the name. Do I have still? No, I thought I have the project name, so I could have done that. That is usually what I do. When I have the reanalyzed data, I use the bioproject names and I put that in the name of my history. So I created my history, I renamed it, and now what we will do is import these raw metagenomic sequencing reads using SRA. So first of things, we need to get these two. So we take the run accession numbers that has been put here, that we put here for you. What you can do is just copy that and creating a new file for that. So please click on copy there, and you go back to your Galaxy. You click on upload, you click on pass fetch data, and here you can pass here your two names, and it will create a new file with these two ID directly in that, and you can click on start. One thing I forgot to show you is also that we can look at the tutorial directly within Galaxy. So if you click on the See Galaxy training materials here, you click there, you are redirected to the training website, and you can again open the same tutorials here. One of the advantage is then the tools is directly interacted. And once you do that, you can click outside and again there, and you are back. And here, for example, Oh, it doesn't work here in this particular example, but you can click usually on the name of the tools and it opens the tools itself. If it has exactly the same in this galaxy, the same version that is required. So now we have our entry, we have our new files there. What we can do is using the tools that is called faster. So faster download and extract reads in FASTQ format from NCBI. And we click that we have a list of SRA accessions, one per line, and it automatically finds our file that we put in our history with that. And we can click on run the tools. And if we go back, it should be that, yeah, exactly what we did. The download can take some time, so I will pause the recording for now and come back once it's done. Is now done. So we have now in our history several files and several collections, and especially the pair and data, where we have our two samples with the correct names, and for each we have a pair, so forward, FASTQ, Sanger, GZ files, and the same the reverse, and so it's what we have for both of the samples here. So the next steps we need to do is to rename these collections as row read. So what you do is you go to these collections, you can edit attributes. You can go inside here and modify it here. All you can do is click here to edit attributes and give it a new names and save it. So now you have a collection that is called row read in your history. Um, so here I was, because it took time, so we gave also an alternative for that. So the first steps after that, so we have the raw read that has been deposited on NCBI. So that's really the raw read that came out of the sequencer. And now we need to pre-process these reads to check for the quality and also to remove potential contaminations. Because the quality and the composition of our metagenomics data sets can really influence the way we build marks and in the process itself. So we need to ensure accurate high resolutions to be sure that we can build correctly marks. We need to process the reads to check for the quality. So it's mean assessing and refine sequencing data to potentially remove low quality bases, remove potential adapters, reads that are too short that could compromise, for example, the assembly. And the second step is to go through the contamination removal. So removing host derived sequences, so for example, in our case, the honeybee DNA, and also potential human contaminations that can then obscure the microbial signals and introduce a bias, especially during the assembly. So we really need to process these reads to be sure that we have clean microbial enriched data that could lead to proper marks reconstructions. So let's dive into these steps. And the first one is to control the raw data quality. You probably have done that. You probably know how to process reads. We have a dedicated tutorials for that for quality controls. But usually the steps we do is to evaluate the quality, trim the reads, so removing removing the low quality basis, usually by a minimum quality score of 15. So every basis at the end of reads that are below these thresholds can be removed. And then once we have trimmed the read based on the quality, removing reads that are become too short. Here we define a minimum length of 15 bases, which is really already really small. And these parameters can be adjusted for your specific datasets and requirements. Maybe you need longer reads, and which may give you probably better quality for the assembly. But maybe you need shorter because maybe your reads are already short. Here we give you recommended value that are the minimum. We think you should not go below that. So, and for that, we will use a tool that is called FastP. And FastP will do these two steps in once. We need already to define that. And then once we have run FastP, so FastP will run for each of the samples and the data together, so the paired reads together, to be sure that when we remove the reads in one of the element of the pair, so for example, of the reverse of the forward, we remove the full full pair. So we keep all the pairs together. So that is important to be sure that FastP is doing it properly. And then we aggregate the results for all the samples into one single comprehensive overview using multi-QC, which give you a web page aggregated results. For that, we will use a workflow that we developed and is available on IWCN Workflow Hub that is called Short Reads QC Trimming. Then, so what you can do is view it on Workflow Hub. So this gives you an idea about the workflow itself, what are the different steps of FastP, multi-QC, and what are the inputs that we will give. It gives you also the ID. So the ID of this workflow is 2025, and we will import it into use galaxy.fr in our case, or in your servers that you use. So to do that, you can directly doing that by launching it directly. So you go to Use Galaxy, and it should download that for you. So you already have this this workflow that has been loaded there directly. So please, you can do that this way. You give it what are the reads. So you see that it directly identifies the raw reads are the paired collection to use as inputs. Do we know that we have adapters to remove the forward reads or the reverse reads? In our case, we don't. We know that we don't have left. You can do that in two steps. So for example, you do first a FastP and it tell you if it has still some adapters, and rerunning the workflow afterwards once you know which adapters you have to remove. In our case, I assure you that there is none. And then you define the qualified quality score. So here, in our case, 15s. That is mean that we keep everything that is above the threshold of 15s. It's really limit. Usually it's 15 or 20, I will say. And the minimal read length. So that could depend also on your input read, this value. Here we put 15s because it's really the minimum. You should not go below, but you can increase this value to be sure to have higher quality, to keep higher quality and longer reads. That will make the assembly much easier. And then you can launch it and run the workflow. And you see that here, when you run the workflows, you have here the 3 input that has been displayed. Here it become green. So it means that it loaded the inputs. And then you have the 2 steps, so FastP and multi-QC. And you see it will become green when it will be launched, orange when it will be launched, and then green afterwards. Here it's overview. You can see the different steps, inputs, outputs, the report, etc., and some metrics. But currently it's loading, so you can keep it this way, or if you want, you can change completely. You can go to something else, doing something else, even in Galaxy while it's running, and still go back to it by looking at the workflow invocation. So workflow invocation will give you all the invocation of every workflow you has been run. And here you see this is the last one we launched 1 minute ago. And you can go back to this page if you do something else in the meantime. Let's go back to the tutorial. So we launched the workflow. I'm sorry, I could have done that also here. Yeah, because it opened a new one, so we can even go here. You see that we still have our in our story, our job that has been launched. So the FastP is running. It will generate a trimmed read and a FastP JSON report. And we can see that we've done that. And I will pause the recording for now while it's running, and then once it's done, I will come back and we will go through the different questions. All has been done, so it's green. If we go back and we see there here, and there is few questions here. How many reads are in each data set? What are the lengths of the reads before trimming? And what are the minimum value for average so base pair quality score for forward and reverse before and after the trimming of and filtering? And for that, we can look at the multi-QC report. So the first question was, how many reads are in each data set? So we see here the number of reads. So that was after filtering. Good questions about before. We have this information. So here we have before filtering. So we see that we have 21 million reads, and after the filtering, that were the combinations. Yeah. So this is for read one, and then for read two, we have probably we have the same numbers. So which means that in total we have 43 million read and in pair. So that means 21 millions pairs. We have a duplication rate of 2.37%. We have a GC content and 0 adapters. And we see here the percentage of reads. So all reads seems to have passed the filtering. The quality score. So this was before for read one before filtering, and we have the two samples that looks really good. So the length of the reads is here we can see 150 base pair, and the read ones are really good before filtering. After filtering, they look the same. For the read two, we see that the quality is a bit worse than for read one, which is usually expected. We're using Illumina, so the read two, so the reverse is usually worse than the read forward. And after filtering, it looks similar because the quality is still above the threshold of 15 that we put there. So not that much has been cut and removed. Here we see that. And we look at here we see what were the minimum average base pair score given the sequence qualities graph, and we see. So that was here. You see the value here at the end. That is the minimum. So we go through the minimum value here, which is here, and we look at the score here at the really end. So it's 32 for the 16 sample, and for the 98, 3.3. So 33, sorry, 33. And we reported this value here. The value can be slightly different because of the different versions of the tools here. But I think we use exactly the morpho, so that should be the same value. Sorry for that. Now let's remove the contaminations and host read. So metagenomic data sets often contain the non-microbial sequence that can compromise the accuracy and quality of any analysis, and that could include the max reconstructions. So this sequence may originate from the host DNA, so in our case, the ONIB, or external contaminations, such as human DNA introduced during sampling or sequencing. And if we don't remove this, they could lead to misassembly, incorrect taxonomic assignments, and biased functional interpretations. So now we will focus on filtering out both hosts, so B DNA and potential human contamination from our read. And for that, we will use again a workflow that is available again on Workflow and LWC. And for that, in this workflow, what we will do, it will map the reads again, the reference genomes of the bee, and again, then the human. So we will run the workflow twice. So the first time through, again, the bee reads using Batut, and then aggregate the result using multi-QC. So we will do a first run of the workflow that does these two steps, so Bowtie plus multi-QC. And the first time, again, the only B reference, and the second time we will run then again the human reference genomes. So 2 times running this workflow. So first, let's start with the running the workflow. So first, you need to import it. So you can launch it as we did earlier. So you use your Galaxy Friends. Oh, sorry, it doesn't. Should not do that. Sorry for that. Oh, interesting. Let's go back here. Let's go back here. Oh, that could have worked. Go to use Galaxy. No, sorry. What I can do is doing from there. So you can do that. Another way you can import a workflow. So if you view it on Workflow Hub, you get this version here. So 2026 in our case. And then you can import this workflow using directly the ID. So here you go to Workflow, you click on Import here. You go to TRS ID. So we know the ID. You click, you give, oh, no, that's wrong, 2026, sorry. And then you have the different versions of the workflow that has been that are available here. So, and then you can import this workflow and add your workflow in your potential workflows. And then you can run this workflow. So that is the two way to import a workflow. And here, so what we want, so we want to bring this workflow on the short reads with the trimmed reads that are in our history. So this list of pairs here. And the host contaminations, so we want to remove the B, and I always forgot which version, so we want the HappyMail 3. HappyMail 3, Apis mellifera 3. So that is the one we want to use. So that is the latest version of the ONIB genomes, reference genome, sorry. And we can launch it. So again, it will give you an invocation of this workflow. It put the data in your history, and we need to wait until Bowtie and multi-QC are finished. Oh, we can already launch the second time the workflow, even if the first workflow is not done, because it will wait until this is ready to launch the second one. So let's launch the second one, and then once we will be done, we can answer the questions in the 2 times. So, okay, we say that first, because we're running, we want to rename that without B. So can we do that? No, we cannot yet. We can do that later. So let's run the second workflow, second time, and afterwards we will rename that, because this one are the reads without the B reads. We will rename that when it's done. So we cannot, we cannot rename it for now. We will rename later, and we launched already the second time the workflow on this one. So this one is again, this one it detected this one as an input, which is the correct one. You can see it's the one we want now. And then the second time we want to use the human, and which versions we want to use is HG38. So this one we want to use on Bowtie, and then we can launch the workflow. It will wait. So it launched already. So you see that we have new data sets or news in our history. It waits until this one are finished. So until this Bowtie is finished, and then it will launch the second workflow. And I pause again the recording until these two steps are done. Workflows for host and contamination removal are done now. So you should have several elements in your history, and 2 extra multi-QC reports, and 2 two collections with that are called reads without host or contaminant reads. So this one and this one. Don't worry about the numbers if those numbers are not exactly the same as on my screen. So the first things we need to do, because they have the same names, is to rename them to be sure that we know what we are talking about. So the first one needs to be called read without B reads. So this is the first one here, and call it read without B reads here. And the second one has to be named read without B and human reads. It's what we did in the second steps here. So please do that and save it. So you should now have these two elements in your history. And we have several questions. So which percentage of reads mapped to the ONIB reference genomes and how many then reads has been removed? So the first things we need to do is then checking the multi-QC report after the reads without B reads. So that will give us the numbers, the percentage of read that has been mapped to the B reference genome. So in these samples, we see that it's 2.8% of the read that has been mapped to the B reference genomes, and on the second sample is 1.1. So we could then remove this amount of reads from that correspond to reads from the hosts. So that is for the reference genome for the B reference genomes, and for the human reference genomes, let's check the latest multi-QC report, and we see that in both cases it's 0.1% of the read, so almost no read that mapped to the human reference genomes, and that being then removed. So it's meaning that the quality is quite good there. So now we have done the first steps for preparing the read before doing the marks. So to be sure that we have clean, high quality read that are mostly focused on microbiome sequences. And now we can move to the steps of assembling the read and then reconstructing the MAGs, so the metagenome assembled genomes. And for that, we will use one workflow that do a lot of different steps. So it's like a really complex, so building MAGs require a lot of steps, and especially to be sure that they correspond to near complete or complete microbial genomes. So this is a simplified version of the workflow. I will show you later how the workflow looks like. But the idea is we start from the sample that has been prepared for quality control and host removal, so exactly what we have done. We could do an extra step of sampling and grouping, but we don't do that today. And then we do a step of assembly with MegaHit here, or Metaspace, depending on the assemblers that we want to use. Here we will use MegaHit, and that will combine our reads and build contigs. So contiguous sequence of reads, so genomic sequence here. We can check the quality of that context using Quast. Then we will do a binding. So binding meaning grouping the context into beans, so discrete genome beans. Beans is like, yeah, genome context that looks the same and that could belong to the same species. And for that, we will use four different tools: MaxBean2, SemiBean, MetaBat2, and Concoct. And for each of them, we will get a list of beans. And then we will use a tool that is called Beanet to improve the quality and the completeness of the beans and combine the beans identified by the different binders and put them into a set of beans that will then correspond to what we call MAGs. Yeah. Then we can pull the beans from the older samples together and do dereplications that will cluster the genomes based on the CheckM2 quality metrics to eliminate any redundancy that we can see between the different samples. to obtain the best quality beans and mags. And then we can do abundance estimations using cover M, then go back to the different samples and identify for each of the beans how many do we see for each of the how abundant are they in the input samples. We can annotate them using GTBTTK, so for the taxonomic assignment. And for the functional annotation, we will use Bacta instead of Roca, that is displayed there. And then the quality will be done checked by Quast and Checkm 1 and 2. And all the report of all these tools will be combined into a multi-TQC report. So that is the overview of our workflow. And so this workflow is available again on Workflow Hub, and it's this number. So I recommend you to do the same as we did before. So do here TRS ID, and you give the ID. So 1352. You take the latest version, which is here. You import that. It say that it imported, but some steps in this workflow has validation error. So it means that maybe there is one tool that doesn't have exactly the same version. No, it means that the server here, so the Galaxy Friends we use here, might not have exactly the same version of the tool that were defined in the workflow. And for that, we need to edit this workflow before running it. And it should show you which tools is not there or which version is not there. So here we see in step 40 that we can use the version 1.10 instead of the version 1.20 that has been specified in the workflow. And here we say, okay, we want to continue with that. It's not a major change in the workflow. And then we save it. That is if it has an error. If it doesn't have an error when you import the workflow, you don't have to do that. And to show you how the workflow looks like, so you see here, so here you have the input, you give the inputs. Here is the steps for the assembly. Depending on which type of assembly you choose, you have different weight or different, you have a branch with Megate or Metaspace. And then everything is combined in Quast. Then we have the binding here that happens for SemiBin. Then here for Concoct, here Metabat, MaxBin too. And then everything is refined to combine with BNet. We have a checkm2 here then because here we have the dereplications with checkm2 running and grep here. And then we have the annotation happening on the bins with GDBTK and Bacta for the functional annotation. And then the quality happens here with Quast, checkm, coverm, and everything is combined in multi-QC. So that's the workflow, how it looks like. Let's run that. So you can click here on run if you are in the workflow editor. If you are not in the workflow editor, if you manage to import and without errors, you click really quickly on here on run workflow. And then the interface of the workflow should be should open. Here for the trimmed read, you'd select the read without B and human reads. It's the same. So here is this case if you have grouped samples. It's not that case, so but just choose the default here. Then the assemblers, we could choose Megate or Metaspace. Here we decide to go for Megate because it requires less computational resources, but it usually generates good quality but shorter context compared to Metaspace. Here we can decide the minimum length of the context to outputs. So everything that will be below the value here will not be kept in the next steps, and it works only for Megate. So here we put a value of 200 base pair. Then we need to provide for SemiBin. So SemiBin is one of the binaries, and it needs It's a machine learning based binaries that have pre-built models, and some of the models are there. So you have the list available here, and when you don't have your environment available here, you can put global. Then you have the minimum mark completeness percentage. So how much your marks, you want to keep the marks when you compare to reference. So if you have a reference, a close reference, you want to keep here, we want to keep everything that at least 75% similar to something available in the reference databases. I think we have a bit of the explanation here. You can have a bit of explanations of how to choose this value. I recommend you to read that when you do the tutorial on your own or when you do your own analysis to choose which value to select here. Then here it's the contamination weight is used by BNet to score the beans. Then here you have the read length, which is used for the coverage. And if you remember in the multi-QC report of the quality control, we saw that the read length is 150 base pairs. So please use this value here. Then you need to select the CheckM database. Please select the one available on your server and the latest one. So here is this version in this server. Then you need to provide the minimum mag length for the replication. Then the ANI threshold for the replication. So the ANI, you have an explanation about here what is ANI, so the average nucleotide identity, which is a measure of the genomic similarity. And in this case, so we want to keep everything, so we want to combine everything that have an ANI higher than 95%, so that will represent the threshold of a species. So everything that is above this threshold will be kept together. So we define that imply that our marks will represent species level. You can change this value if you want to have strains of the same species. So you can put a value of 100 if you want. And if you want, here you have an explanation of the different value. What does that mean, the different value at ANI, and what are the applications? How we to use that? And which threshold using here, you have a bit of explanation not to select the best threshold. So in our case, we will choose the 95% because we want to dereplicate at the species level. Then here, the next value is the maximum max contamination percentage. So everything that will be below the threshold that have so this value is used by DREP, and so the threshold will be used to define the percentage of contamination you want to keep. So the default by DREP is 25%, so let's keep that. But here you can, for example, you can use a value of 10%. Then the next element is do we want to run GTBTK on the marks? It can take a long time, so if you are interested, I usually do it because I want to check my marks and the taxonomy assignation of it. But yeah, that keep in mind that can take off time. Everything takes time, honestly, in this workflow. So yeah. And then you need to select which database, and you need to select the database based on what is available on your server. So here it's this version for me. Then the Bacta database, please use the latest one, same for the AMR Finder Plus. And then once you have set up all these value, you can click on the run the workflow. And then it should again launch this invocation graph. So you can see again the workflow we have and with the different steps, and that will become green when it's running. I will do a quick stop there while it started, and then we can continue because we prepared the data already for you to wait to continue the analysis, because these steps definitely, this workflow, not just this step, all these steps takes time, and especially the assembly, the binding, the annotations, that can take several hours. So you can launch, you can see, you might have different results than the one we will see later, but that should be in the same range. But keep in mind that takes time. So you can pause, wait for that to be run and do something else in the meantime, or you can follow following the next steps using the history that we make ready for you. Running the workflow take a lot of time. So what we can do is import history with pre-computed result. So for the context, this history has been generated 7 months ago, so the result can be different now of running the workflow. It should be not big differences, but it could have been numbers can be different from what we see in the workflow, in the history, and in the result you can see in the tutorial itself. So if you have similar but slightly different result, it should not be a problem. If we see big differences, then there might be an issue that we need to resolve. So let's import the workflow. So please, whoops, this one doesn't look. Okay, interesting. And if I do that on you, oh, it should work. So if you do that, you go there on the history, and then you can import. You can click on switch to this history, import and switch to this one. So I will use this one in this case. So that is on EU, on the European server. You can see I've been imported on this one. So that has been generated in December 25. So as I said, the result can be slightly different. Where did I go? Let's go back here. No, assembly. I was there. So we have imported then the workflow here. You should have a lot of steps added to the workflow. It should be around 52 gigabytes in your history. And we have a lot of results that has been generated. So the first steps to do we do when we do the building maps is to do the assembly, so reconstructing genome sequencing from metagenomics read. But this is a complex step because it's like trying to solve a jigsaw puzzle, a complex one, where the idea is to identify reads that fit together by detecting overlapping sequences. But unlike traditional jigsaw puzzle, this task is complicated by several challenges inherent to genomics and metagenomics. First, the genomic complexity. So in genomes, we have repeated sequences, we have missing fragments, some errors have been introduced during sequencing that make accurate reconstruction difficult. So we don't have exactly the same matching sequences that can be combined together. The data volume is a second one. The metagenomic data can be really massive, and that requires significant computational resources. For example, for megarit or for metaspace, we need several, it can be tens or hundreds or thousands, maybe hundreds of gigabytes of RAMs to run the assembly. That depends on the complexity of your sample. Then community diversity. We have unequal representation of the different member of the microbiome community. That could be dominant, where that have really abundant sequences for that, and we can have really rare species. So that means we have only several reads for that, and that make really difficult for doing the assembly. Then we have genomic similarity. So if we have closely related microorganisms or multiple strains of the same species in our sample, then it really adds the complexity there, really different layers of complexity. Then data limitations. So minor community members might be underrepresented that can lead to incomplete or fragmented assembly. So for that, there is a variety of metagenomic assembler that has been developed, including Metaspace and Megate that we offer in this workflow. And they have all their unique computational characteristics and performance that can vary depending on the microbiome that has been studying. And so there is initiative like the critical assessment of metagenomic interpretation that try to benchmark in a global way the different assemblers. And you can recommend you to look at the different papers to have an idea about which assembler choosing in which context. Depending on if you want to maximize the contiguity, if you want to maximize accuracy, if you have to have computational efficiencies. And this is really essential for achieving the high quality assemblies. If you want to learn more about assembly, about algorithms and the tools, we have a dedicated tutorials for assembly of metagenomic sequencing data. So in this workflow, we run Megate and Metaspace, but Metaspace could be also an option. So let's now look at the results. So how many contigs were assembled for the 98 samples and for 76 for the 60 samples. So for that, let's go, oops, sorry, not this one, so this one I can close. For this one, and I will go slow scrolling down in our history to go until we have the assembly here. So here we have the result, and here we can see how many samples has been rebuilt. So for the 98, we have 534,000 sequences. Then for the 16, we have 572,000 and more sequences. So contigs, yeah. Yep, yeah. And what was the minimum length of the contigs? So if you remember when we selected the parameters here up there, we say that we want to have a minimum length of 200 for the different contigs. So that was so all the contigs have a minimum pair length of 200. And if you look here, we see that the length is 236. So if we open, we will see nothing. Here is the length of the contigs. So here we have the length 200, here 240, 202, but we will not see anything below this value if we scroll down. So once assembly is complete, we can assess the quality of the assembled contigs, which is really crucial to ensure the reliability and accuracy of the downstream analysis. And this can be systematically evaluated using MetaQuast, which is a metagenomic mode for the Quast tool. And this tool provides a comprehensive suite of metrics to evaluate the assembly performance that include the contiguity statistics, such as the numbers of contigs, the total assembly length, the N50 and L50 values, which give an idea about the length and distribution of the assembled sequences. It can also do evaluation analysis of the genomic features, including genes, operands, to assess the biological relevance, and can do comparison to the reference genomes if available. And so by using that, we can really get a really good insight about the completeness and accuracy and overall metrics of the metagenomic assemblies. So Quast generate multiple outputs for the main, but what is we are interested more is the HTML reports with the different metrics. What we will do, we will open the metrics. So where is it? Quast. So here we have the Quast, we have the tabular reports, PDF, tabular, and here we have the HTML report with two reports. So one for each samples, and we want to check them side by side. So we will look using the window manager. So click there, you should have a small cross here. And then here, if you open that, you see that it doesn't appear on the middle, but on the top. And so you can see the two samples side by side. Here, the two reports for each sample side by side. On the top, we see we have a summary of the result, and we will spend a bit of time on that once. So first, what we have is we have the statistic without reference. So please click also on extended because it gives you more details for each of them. So statistic without reference, so the number of contexts. Here we have the numbers, then we have the value here. So we have a value that sounds different from the one we saw initially, when we look at the FASTA file. It's because Quast here doesn't report only the context that are, if I remember well, higher than 500 base pair. So that is the default for Quast. Quast here in the number of contexts report everything that is above 500 base pair and not everything. So everything is here, you see. Here is the same as what we saw before. What is the length of the longest contig? So longest or largest is this one and this one. So in this sample, it seems longer than in this sample, if I'm correct. Yes. Then the total assembly length is this one. If we take only the sample that are the contigs that are longer than 500 base pair, is this value, and this value is for all contigs. So here the value. So even if we have a largest here and more contigs, it seems that in total length, this one is bigger than in this sample. Then how similar are the microbiome community between samples based on the context? So the two samples exhibit complex statistic. So they have similar numbers, but they have longer, as we said. So we need to look a bit beyond that. So beyond simply context and measuring their length, there is two statistics that are essential. It's the N50 and the L50. So N50 represents the length of the shortest context where the combined lengths of all contexts equal or exceed half of the total assembly size. So the higher the N50 is, the better contiguity there is. So meaning fewer gaps, longer contiguous sequences in the assembly. And the L50, the minimum number of contexts needed to cover at least 50% of the total assembly length. So a lower L50 suggests that the assembly is more contiguous, as fewer and longer contexts are required to reach half of the genome total length. So here you have a better explanation, or better understanding of what is N50, L50 with examples to try to explain them. So what is the N50 for both samples and the L50 for both samples? So N50 here is 2,213 and here 1,751. So it means it is better on these samples compared to these samples, and the L50, so we need here less context, so this one is better than this one. So as we said, the length, it was a bit what we saw there. So despite the largest context to be smaller here, all the other contexts closer to this one seems to be longer in these samples compared to this one. So yeah, it's more contiguous on this one, in these samples with longer contexts covering half of the total length. So both assembly are fragmented, is mostly better contiguity overall. Yeah, that could reflect variation in microbial diversity, sequencing depth, or assembly performance. So then another step is the read mapping. So read has been mapped. So we use the reads and we read. So the reads has been used as an input also for Quast. So the read that we, without host and bee, so bee and human reads. They are mapped on the context to see how much of the read has been used to build the context, how much read were discarded because maybe they couldn't build context. So that is a read mapping step here. So here we have the percentage of reads, the number of read that has been mapped, and here the percentage. So we have 97% here of the read that mapped to the context, and we have 95% here. So that's interesting. So it means that this is the number of read or percentage of read that has been used to build the context. So that is the first report that we got for Quast. Well, that is the first steps we do for the binding. So globally, the binding, it's not the binding we are looking at, the assembly. And we already have statistics for that. So it seems that the assembly went pretty well. We have quite a big number of contexts. We have good N50, L50 values. We have a high percentage of read that has been used to build the context. So globally, the mapping, the assembly was good. So now what we want to do is doing metagenomic binding, which is the process of classifying DNA sequencing from metagenomic data into discrete groups or beans based on their similarity. So the idea is to assign sequence that will correspond to same organisms or same taxonomy groups, to then use that to define the marks. So marks will be, or a bin will be a set of contexts that could belong to the same species, and that could be the basis of a genome there. So it's binding relies on the combination of sequence compositions, coverage, and similarity to group the context into bins. They ideally represent genomes of distinct microorganisms present in the samples, but it's a challenging process because of the genome complexity, as we said, because of repeated sequences, strain level variations. We have also the issue of uneven sequencing coverage across the different microbiome species, and we can have also contamination in chimera that can be built during the assembly. So that could be also bias the binning. So numerous algorithms have been developed for metagenomic binding that all have their own strengths and limitations. And the choice of the tools to use depends really on the characteristics of your datasets and what you want to do. There is good benchmarking that has been done, especially with the CAMI initiatives, that could provide a good guidance on selecting the most suitable binders for your specific datasets. You can also read more about and doing it on your own with the binding tutorials that we developed. But really a widely adopted strategy for building marks is to use multiple binding tools together to really gain from the individual advantage of each and all the methods to then can be used afterwards with a bin refinement to consolidate and improve the result of each of them. So this approach really use the strengths of the different algorithms, produce more robust and reliable set of bins. And for this analysis, so in this workflow, we use 4 complementary binding tools that each of them offer different advantage to capture the microbiome diversity. So Batut, which is known for its efficiency, accuracy on binding metagenic context, MaxBin2, that use probabilistic models to improve the binding accuracy, SumMibin2, which use a deep learning approach to enhance binding performance, especially for complex datasets, and Concoct, which use a Gaussian mixture model to cluster context based on correlated compositions. And then we can really look at the result for each beaners. So we can extract the number of beans and the number of elements for each of the beaners by looking here. So if we go back in our history, in our galaxy history, where I was, here, Quast. Then we have the contigs here for concoct, and we see here is the number. Oh no, that is contig. Sorry, I got lost. Concoct. Let me check. Word concoct, that is called beans. So reconstructed. So semi-bean here we have the reconstructed beans, and here no, that was not what I wanted to do. Okay, let's go. Let's search for MetaBat first. So MetaBat here, and we see we have all the element with MetaBat. So here, bean sequences here. So here we see four. We have now a collections of collections within. So we have a collection, and if we go inside, we see we have for each sample another collection, and in this collection we have FASTA file. So each FASTA file represents all the one bean, sorry. And for example, for this bean one, here we have 133 sequences, so which means these beans correspond to 133 contigs that has been considered by concoct corresponding to the same beans, so potentially to the same species. So here in this collection, we have 35 FASTA files for these samples and 25 for these samples. So here it will mean that for these samples, we have 25 beans and, 34 beans here, 25 for this sample here. And we could put a table together, so it's what we did there. So here for MetaBat 2, we have 35, 25. Then what do we do for, we got for max bean 2, so max bean. Here, the beans is in this collection that is called beans. So we have 45 for these samples, 35 for this sample. Then if we go for semi-bean, we need to go for recursive beans. We have 16 here and 20 there. Then concocted the last concoct. And concoct, we need to go to this one, and we have 80 and 57. So here we summarize here. So which binning to generate the most and the least beans for each sample. So in every case, this concoct seems to generate the most bean. So that means maybe concoct tend to produce finer beans, maybe potentially more fragmented groupings. So maybe it could capture more nuance than with microbiology, but it could be maybe oversplitting genomes. Semi-bean generate the fewest bean, maybe because of a more conservative approach, producing maybe larger but more complete beans, but maybe missing small distinction between closely related microbes. And max bean 2 and metabat 2, they are produce intermediate number of beans, and that could be a balance between granularity and completeness. They seems to have comparable performance. If the trends or the number of beans generated consistent across the different samples, so is it the same in every cases? I think too, we can say that. It seems to be the case. In every case, So we have more beans all the time for this one sample compared to this one. Beyond simply comparing the total number of beans, we could also look at the contigs per beans for each binding tools to really have a deeper insight about the quality and granularity. For that, we will use the collections of collections. So let me check which is the names of that. So I will need to remove that. And we should have something that is called collection. Where is it? Collection and other as a list here. And here if you, oh, is it this one? That collection and other as list. Okay, let's search for that. We have this. So we have then a list of tabular datasets. So this one, this collection contains 2 sub-collections, so one for each samples. And within each sub-collections, we have 4 tables that correspond to the different binding tools, and each table consists of 2 columns. So if we saw there, we have 2 columns with first the names, sorry. Here, this is a, I need to disable that first. We want to see that in the middle. Column 1 is the name of the contigs, and here is the name of the beans where it belongs. And so what we can do is can group the contigs by their ID number. So on these collections in things, so it's the same for each of them. So for example, here I think it's the first. The first is metabat, then maxbin2. So here, metabat, maxbin2, semibin, and then concoct at the end. And so what we do, we want to group by the numbers to be able to count here the number of sequences that we have there. And summarize that afterwards to have generated tables for each sample that contains statistics of the numbers of contigs per beans. So let's do that. Okay, I will go, oops, wrong tutorials, contigs, tutorials, whoops, microbiome, reading here. In the binding here. Why I do want that? Because I want to use this specific functions that open the 2 tools with exactly the same version that has been run. So here we want to use the and others as, that was a collection. And here, please, and others as a list. So this is a collection that we want to use. This collection here. And then what we want to do, we want to group by column 2 in each element of the within these different collections. And then in this, we want to do an operations of counting, count on column 1. Yes. So we want to group by the column 2, and then knowing number of lines for each. So that will run on the top of our history. And once we have that, we can also run, oh, why is it going back again on the top? Here, statistics summary. And that will run on the group collection. So once we have done that, we want to get the summary, and the summary is column or expression. So we want a summary on the column 2. So the column 1 will be the name of the contigs. The column 2 then will be now the number of sequences for each of the contigs. So the number of contact, no, sorry, the column 1, sorry, will be the bin ID, and the column 2 will be the number of rows that has been found for this bin, so the number of contact that has been assigned to these specific bins. And here we want to compute statistics on that, so mean, yeah, for example, yeah, the sum, the mean, the standard deviation, and then the different quartile values. So we do that, we launch the statistics, and then we want to collapse the collection, so to get only then one element or one keep one either line. Yes, we want everything as one table and not two tables. Oh, we want. No. What we want here, you see, we will have 4 tables, so for each of the tools, and we want to group for each samples all the tables into one to get then one table for each sample. While it's running, I do a small break. We have now 2 tables, so a collection with 2 tables. Oh, what's going on? Interesting. So we have 2 tables, one, and we can enable again that here, and these 2, and we can look at the statistics. So what are the statistics of the number of contacts for each year, right? But again, the numbers that we have, we should have the similar ones here. So first rows will be for concoct, metabat, backspin 2, and semibin. So that is the order that we put that here. Then how do you interpret the binding statistics tools for the samples? So it seems that in every cases concoct used. So here we have the mean. So how many contacts are in average for each of the bin. So in both cases, it used quite a lot, but not the most. The most will be this one, which is max bin 2. Okay, let's go there. Let's compare here. That will be easier to compare. Use more context to generate the bins with a wide range of context per bins. Though that could explain that context create many small bins. I have just a doubt here. From here. Okay. Nope. Here, Metabat2 use fewer contexts and show lower variability in the number of contexts per bin. Then bin 2 use a moderate number of bins, but have a higher mean numbers of contexts per bin. So this deviations is quite high, so indicated variability in bin size, and semi-bin use the fewest bin, so fewer contexts and show low variability in context bins per context counts per bin, sorry. So we have then discussions about the distribution. So with the median being here, so at 18, big higher for the others, especially for max being 2. So it's highly skewed. So distribution is highly skewed if you go from here to here. So this could suggest potentially oversplitting context into many small bins. Same for both samples. For the Metabat2, so it's a relatively narrow interquartile range. so even a more even distribution compared to concoct. Max bean 2, so we have a broad distribution of the values, so reflect to have larger beans, so beans with many contigs, and semi-beans, so we have quite a uniform binding process. So in every cases, concoct and max bean 2 produce more bean than metabat and semi-bean, but the distribution of contigs per beans is more variable for contigs. And the sample, so this sample has largely slightly more beans across all tools compared to the other samples. So that could reflect, so here, that could reflect a difference in diversity, microbiodiversity. So what are the last question? What are the practical implications? So concoct might be useful to capturing fine scale diversity, but needs additional refinement to match small and fragmented beans. Max metabat 2 and semi-beans are likely to produce more variable and consolidated beans, making them more suitable for downstream analysis like genome reconstructions or functional annotations. And Max bean 2 offer a balance between the number of beans and the number of contigs. The next steps we can do is to evaluate the bindings using CheckM2. So CheckM2 is a software tool used for metagenomic binding to assess the completeness and contamination of the genome beans. So it compares the genome beans across, it compares the beans that are generated to a set of universally single copy marker genes that is supposed to represent nearly all bacterial and archaeal genome that we know right now. By identifying the presence and absence of these marker genes in the beans, it can estimate the completeness of the genome beans and the degree of contaminations. So if we look at the workflow, we see that we run CheckM2 here, but only at the end, so after the refinements. Here, I just wanted to show you how the quality by running it in an additional steps. Oops, where is it? Running, yeah, the workflow, a workflow that has been developed for that, so that is available as a public workflow on the server, so either on org, on FR, on EU, or on the Australian server. And that's run CheckM2 on the different beans that have been generated, and then I aggregate the result into with multi-QC. So that is this workflow is. So please, so first find the workflow here. Yeah, you can disable that, go back to your history. Then in public workflow, if you search for quality control, you should be able to run this workflow. Should be able to, that is a public workflow. You can directly use it. You don't need to import it. You can use it. So it first asks you the metabat2, so metabat2, and you need to give the bean sequences. Then max bean, you give it max bean 2 these beans. That was selected. Semi-bean, reconstructed beans, and concoct. Here you give it the beans. And then you can run the workflows. And so as you see, this workflow, what it does, it just flat the collection, extract element identifier. It's just to extract the names of the samples, then doing relabeling. So it's mostly a reorganization of the things first. And then it merge all collections, running the CheckM2. So this is just pre-processing and preparing the samples here by adding yeah, it's renaming the files within the collections. Then we merge everything to have one collections with all the bins from the different binners all together. We run checkm2. So here we add, we say that it's a metabat2 and plus the name there. We merge everything to have one collection with everything. We run checkm2 and then multi-QC. It will take few minutes, a bit more to run that. So I pause again the recording and start again when it's the quality control of the bins has been finished. So checkm2 and multi-QC has run, so we can check the result. So we have one multi-QC report where we can see for each of the bins here, we have different information. So bins quality. You have one table here where we have first the sample name, the predicted completeness, the contamination, the coding density, the average gene length, the genome size, the gene C content, and then for each bins we have this information. And you see you have concoct underscore the name of the sample and the number of the bin here. And you have that for each. So concoct. If we scroll down, here we change, we have the different sample, and then we go to max bin 2 and same information there. So that's how it looks like. So what we can do is looking at how many bins have a predicted completeness of 100% and greater than 90% for each samples. So what we can do is we can sort by the predicted completeness, and we see that here we can count base. So we have 1, 2, 3, 4 for the sample 98. So 1, 2, 3, 4, 5, 5 for 100%, and then 5, 6, 7, 8, 9. Okay, 9. Where I was. So 1. Okay, we say 5, 6, 7, 8, 9, 10, 11, 12, 13, 16, and 14, 15 for the 98 sample. And that's what we get. Yeah, but I had to split between binars and that. So you have the result here. So we could we could check by calculating for concoct how many we have, and we have a lot for 100% with a good completeness, really good completeness. Metabat has several, also with a good completeness. Then we have different numbers here. So concoct we have 4 with 11 with high completeness of 90% for this sample, and then again different values for the other sample. And so concoct seems to produce the highest numbers of high completeness bins for both samples. That suggests that it's more effective at reconstructing near complete genomes. However, all binding tools except semibin contribute to the high completeness bins. Yeah. So yeah, it's mean that semibin we don't have any bins that are really good in semibin in both samples, and the others provide also good quality. That is Metabat, Backspin, or Concoct. So by combining the different binding tools, we could recover good or complete or near complete microbial genomes. But the second things we need to check is the contamination, because if you see here, we have near complete genomes, but we have a really high predicted completeness. So the next question was, how many of the beans have the predicted completeness greater than 90%? That have a completeness greater than 90% have a predicted contamination above 50%. So if we look there, so all this one have a 50 over 50, this one also have above 50, this one has a really high contamination, this one also, so really a lot of them. So if we look with the number of bins above, with the completeness above 90%, that have a contamination above 50%. So for Metabat, it's 0 above 5. So all the 5 bins have a good contaminations. For this one, it's 1. This one over 2 has a bad contaminations. This one is to keep. Yeah, here we have 6 of the 11 bins that have bad contaminations, 6 over 9. So it means that a significant portion of the high completeness bins from Concoct exhibit really high contaminations level above 50%. So while Concoct effectively capture incomplete genomes, it might also include substantial contamination from other organisms that might compromise the accuracy of the analysis afterwards. Metabats and MaxBin produce fewer contaminated high completeness bins. And so really that suggests we really need refinement strategies after the binding. So that's the next step. So the bin refinement. So to improve the quality and completeness of the bins. As we see, the binders have produced different beans that could be fragmented, so because they are not complete, they can be redundant, so providing the same mean are beans, and they can be contaminated beans. So the bind refinement is really a critical step there, and it aims to improve the quality and completeness of marks by removing contamination from beans and merging related beans that might have been incorrectly split by individual binding tools. So for this workflow, we use BNet, which is the successor of MetaWrap. And so BNet leverage advanced algorithm to automatically detect and remove contaminations while merging beans that likely originate from the same microbial genome. And so what it does, it integrates results from different binding tools and then improve the accuracy and reliability of the marks, make them more suitable for downstream analysis. So BNet generates several collections, one with the conserved bin and collection of quality reports. And so what we can do is looking at the quality report. So if we look for BNet here, so it's what we see. We have 2 final quality report, and bins. And I think we need to open the final quality reports. For that, we to see them side by side, we again open using the window manager, and we have there the 2 report. And what do we see there? We see here the name of the bin, its origin, the name, the completeness, the contamination, a score, the check-in model, size, etc., etc. So how many bins are left after refinements that we might not need that. We can see here the number. So we have here timeline. So 9 bins have been kept for this one. And here 5, so 4 are kept. What are the different columns in the report? You can see the names here. So the name, origin, original, etc. And if we want to really see what are the different columns, I recommend you to go to the BNet GitHub repository because they have an explanation or they have an explanation here of what are the different names and what do they mean here, and we copied them here. So that indicate the source, if it's original, etc. So what is the minimal value of the completeness for the refined beans? If we go there, please, why are you doing that? Here. The completeness, the minimum value was here 77, 76. And if you remember, when we defined the workflow, the value of the workflow, we had a minimal completeness percentage of 75. So that was the value. So that's why we don't have value below 75 here. Bin refinement. How many bins have a completeness greater than 99? And what does that indicate? So we are here, one bin here, and one bin here. Oops. 3 bins plus. Yeah, it's true. They are not all. So 1, 2, 3 bins here, and 1 here. So it indicates that these are near complete representation of their respective microbiome genomes, so which might be high quality genome reconstructions. What the next value is, what is the maximum contamination? And if we look, the maximum contamination is 7 here, and it seems to be 7 also here. So this value indicate that some bins still contains a significant portion of sequence from multiple organisms, but that is much lower than what we had before that. So the refinement really improved a lot the quality of the bins there, or at least the bin that has been kept. And the next steps we can do is doing a dereplication of the max that to reduce the redundancy by identifying and retaining only the high quality marks. And so what we did, we did individual, we have different samples here that could represent, if we look there, different abundances because of different sampling time, different conditions. We did the individual assembly and binding and refinement for different samples, and the replications consist of grouping everything, so the bin for all the samples, and then doing a replication to combine bins that could belong to the same organisms from the different samples. But it needs different questions that need to be thought before. So what are the thresholds we want to define marks? 99%, for example, or 95 of average nucleotide identity. In our case, if you remember, we could choose 95% to keep at the level, the species levels. How do we determine which marks is the best representative? So for each of the bins, so which is the best one? It's usually based on the completeness, contamination, and assembly quality. How to ensure that the final base mark set is both representative and redundant? So to maintain redundancy in a mark data sets, so maintaining this redundancy can lead to challenging the steps of mapping reads to marks, where sequencing reads might align to multiple high-quality marks, and this can result in artificial low abundance estimate for each taxon, misinterpretation of population dynamics, and complications in curations, for example, with Envio. So here, the studies have applied various similarity threshold for the replication, including a 95% averages. You can see the paper here, 98 or 99% of here amino acid nucleotide identity and not average nucleotide identity. So again, you can read again about what is ANI and what does that mean. And so for the dereplication, we use a tool that is called grep to cluster marks based on the sequence similarity and retain only the highest quality representative from each cluster by incorporating CheckM2 quality metrics and making sure that we get marks that are non-redundant and optimized for downstream analysis. So in our workflow, we use grep from the bins generated by bnet across all samples. Here we only have 2, but if you have more, you will pull all together from, and then CheckM is used to evaluate their quality. And here in our case, we have selected several criteria like the completeness of 75, the mag contamination of 90, the minimum mag lengths, and the ANI dereplications. So what we can do is now looking at how many bins are given as input to grep and how many has been kept by grep. So if you remember well, we have here. So what has been used by grep? So we gave it how many bins? So 9 for these samples and 4 for this one. So 13 bins has been given to grep. And how many have been kept afterwards? This is, where is it? I think it's this one. It's a good question. Where are you? Dereplicated. So we have then a list of 10. So 10 are kept afterwards. So that is the number of the dereplicated genomes. If we look then on the primary clustering, so that is the tree we can see here. So here we have our 13 samples, and we can see how there has been merge. So the merge happen for everything that is before this line here. So based on the ANI here of 95%, that should be somewhere here. We can see that these two samples, these two bins has been merged together into one. This one here, this one here. So it's like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. These are the representing the different bin that has been kept here. Yeah, that is what we try to explain. So then we have high confident non-redundant marks. So we have 10 marks that have been generated from our data sets. So now we want to really look at the quality more in details of these marks. So we have set the completeness and the contamination of both using CheckM and CheckM2. That so there are two tools. So there are two versions of so CheckM2 is a successor of CheckM. CheckM use a database of lineage-specific marker genes, while CheckM2 use a machine learning approach to improve accuracy and speeds for novel species. So here we have a detail between the difference between the two. Here we run really the both of them, honestly, to prove, because they gave really slightly different results. So the question is there, how many bins have a predicted completeness of 1% and greater for CheckM2? And for that, we need to search for CheckM. Quality report should be this one. Yeah, it's this one. And what we see, how many beans have a predicted completeness of 100%? 0 completeness. And how many beans more than 90%? 1, 2, 3, 4. 4 beans are, I put 3, but there must be a mistake here. So it's 4 here. And what the different marker, lineage, and taxonomic levels found by GTN, and not GTN2, but GTN. Let's look at that. I will disable that. GTN, where are you? Bin, oh, yeah, no, that is a check. Here we see the marker lineage in the second line here, and the different marker lineages. So if we group them, we see we have 2 bacteria here. We have 1, 2 enterobacteria. I cannot pronounce it, sorry. I will not even try. For the family, then here order, we have 1, 2 lactobacilli. Here we have proteobacteria. Here we have 2 gamma proteo and 1 clostridiale. So that is the numbers. So we have from the kingdom, then classes, we have 2 classes. So that's what the highest they could identify for the marker, for the beans and here. So it gave us already an idea about the quality of the marks. And the next step was to identify the microbial abundance estimation from the marks. So we have the marks, we build marks. We have genomes, and now we would like to know from the raw reads how many reads or what is the abundance that we can find for using the reads mapping to the marks, giving an idea about calculating the genome coverage statistics. So the genome coverage is determined by summing the total aligning basis across all contexts on a mark and normalizing by the total genome length. And that really gives us an idea about that and will give us idea about the per genome coverage statistics. So for that, we need to look at the multi-QC report. That should be this one here. No, the multi-QC report, so the web page here. And here we have a lot of statistics. So we could also look at the BNet. Oh, I could have looked directly there. That would have been much faster. So the GTN2, we already have the result here. I don't know why I didn't open the GTN, the multi-QC. I forgot about that one. So then the cover M here. So what does the relative abundance represent in this? What does it represent? So it's proportion of sequencing reads mapping to this specific bean. So for this bean that has been originated from these samples, we have for these samples 2.3% of the read mapping to it and 0.3 from these samples to this bean. And for this one here, we have 2.7 and 2.0 here. So what are the percentage of unmapped read for both samples? So it's 92 and 97 here. So it's a quite big number. The unmapped reads, what are these unmapped reads? So they represent sequence that could not be aligned to the assigned marks. This occur because they correspond to uncharacterized microbes. They can be non-microbial DNA, but I think given that could be something maybe from leftover from hosts. It could be technical limitations or incomplete assembly. So that you remember we had really good quality the assembly. So the number of read mapping to contigs after the assembly were quite good, 90 and similar numbers as we have currently with the unmapped reads. So we had assemblies, we had good quality assemblies, but they might be too fragmented to build marks from them, these assemblies. So it's can probably because the beans were not good enough to be kept as marks that we lost this number there. We could improve that by reducing, for example, the contamination level or the completeness that we want. So yeah, reducing the percentage we put there. So the 75 and then 20 and the 50% there. We could change that and that will change the value. What is the relative abundance of this? So the being 6. So it's 2.7% and 2% in this sample. So that indicate this organism. So the corresponding species here is similarly abundant in both samples. And for the bean 2, as we saw before, the difference is really different. So that means, so this species is 8 times more abundant in these samples compared to this sample. So that could suggest that there is environmental conditions or the biological interactions or something specific that may favor the growth of the corresponding species in these samples compared to these samples. That also indicates a shift in the community structure between the two samples, maybe because of the difference of treatments. It could be something different here. Why do some marks show a significant difference in relative abundance between two samples, and what could indicate? As I just started to mention, it could be environmental and biological variations that could explain the differences. It could be sample-specific microbial populations. It could be technical factors. So, for example, differences in sequencing depth or library preparations. It could be biological competitions of interaction. And it's where replicates is important, especially to decipher the technical factor from environmental or biological factor. So if you can, you see in sample, in replicates from the same, exactly same experiment, from two experiments or two treatments, and all the replicates have the same differences between the two treatments, then it's come from the environmental or biological differences. But if you see differences because within a different same treatments, that could be a special sample specific, but that could be a technical factor. So that you need to keep in mind that while you interpret. And then when we see that, we have already a lot of information here. We start to dive really more into how to interpret the results. The next step is to interpret, to assign taxonomy to these marks. So it's a really critical steps in metagenomic analysis to really identify and classify the genome that we built now. So we can use that to interpret the microbial diversity, explore the ecological roles, and compare communities across environment. So for that, we use the Genome Taxonomy Database, so the GTDB, which is a comprehensive and standardized resource from bacterial and archaeal taxonomy. It uses genome sequences to define taxonomic relationships, which is different from traditional taxonomy. And it provides a consistent and up-to-date framework to classify microbial genome ensuring that taxonomic assignments are accurate and reproducible. So GTDB include genome-based taxonomy using 120 single copy marker genes to infer phylogenetic relationships. It uses standardized nomenclatures to follow consistent naming conventions to reflect the evolutionary relationships, and as regular update to incorporate new genomic data and improve the taxonomic classification. And to classify genomes using GTB, we can use GTBTK, which is a bioinformatics tool designed to classify microbial genomes using GTDB. So here you have a bit of explanation how GTBTK works. And we already have run GTDB-TK. If you go through your result here, you should see, where is GTDB? So we have results here, we have summary. GTDB. Oh, no, GTDB. We have different, so summary, classify, identity, align. So the summary, if we see there, can be a bit hard to interpret. It's a lot of information, and we might not need all this information. So what we want to do is we want to extract. Here we have, you see, we have one collection with only one element. So what we want to do first is to extract this element. Where we are. Here, extract datasets. GTDB summary. And we want to run to get the first dataset. So then we see we have directly the file here. And then what we want to do, we want to keep only the first and the second column. I think the rest is not really needed. So let's cut column 1 and column 2 on the GTDB, the summary here. That I can close. And the next step is what we want to do then, once we have that. You see that on column 2, we have the taxonomy displayed at the different levels with the domain, semicolon, the phylum, semicolon, the class, etc., etc. So what we would like to do now, once we have this with that, we want to have that in different column. So that is one column, that will be a second column, will be a third columns. Why my cut is not visible? That's a good question. Did I submit it? Okay, yeah, you see. So what we will do now is then, okay, yeah, true. Fair. I reload the page, so we lost where I was. Where are you? Here. Replace text. On the cut, then what we want to do on column, on column, we cannot yet, I think. What we want to do, we will want to do is to replace the semicolon by anti/t, which means a new column. So create a new column there. And I think we need to wait until the cut is done there. And I will do a short break there. Done. So we can now select, we want column 2. And then we can run it. So it will create, as I said, Here we have two columns with the name of the beans and then the classification with semicolon between the different taxonomic levels. And here we will have the different taxonomic level in different columns. And then what we can do is what is the lowest taxonomic level identified in the GTBTK classifications? Have all beans assigned uh, have a label to at these levels? What are the different taxons found at the different levels? And then we can also compare to what is expected for the bee gut microbiota. Let's wait just a bit for that. It takes a bit longer than I expected. Just doing a really quick break, really. So the replace text is now finished, so we can look at how it looks like. So we see now that we have the bin, we have first taxonomic level, or the highest taxonomic level, and then different levels until the species level here. So that is what is the lowest taxonomic level. This is the species level, which is somehow expected there. Because we grouped at the 95%, so ENA percentage, which correspond to the species level. Have all beans been assigned? So most except one. So the latest one has been assigned until the species level. What are the different taxon found at the different levels? So here you see it's mostly removing this underscore, this level underscore, and we see that here, we just display that here. So all belongs to bacteria. The phylum, we see we have, if we group, so I reorganized here to make it more coherent, to me similar. So we see that we have all the monadata here. So we have 1, 2, 3, 4, 5 beans from this phylum. And then we have Bacillota and 1, 2, 3, 4, 5. So it's mostly Pseudomonota and Bacillota. Gamma proteo at the classes. Why? I think it's that is not the case anymore. But yeah, that's a leftover. Then at the classes level, so Clostridiale, we have one classes for that one. And 1, 2, 3, 4 for so they are from the Bacillota. Gamoprotactaria is the only that is from the Bononota. And after, if we go to the order, we have Enterobacterales from the Gamoprotactaria. We have Burkholderial. So I'm really sorry for the mispronunciation of all these taxonomic levels. And here we have Lactobacilli, and then we can go really deep there. So, and it seems that Apilobacillus is the most abundant at the genus, and then we have species there. So we have really a diverse microbial community with different phyla, classes, order, and with different dominants. So the question was afterwards, the only bee gut microbiota is relatively simple. It's dominated by five core bacterial lineage corresponding to cluster within the genera Bifidobacterium, Bombilactobacillus that were previously Lactobacillus, Guilliamaale, Lactobacillus, Snodgrassella. So we have also non-core bacteria that are commonly present, like Bartonella or Frischella. Are the species or the genera that we saw are expected in the sample. So observed genus, so we have Gimelae, and we said and we know that it's expected, so it's a core member. Klebsiella, it's not the case, so it could be a contamination. Seriatae is occasionally because it's a pathogenic bacteria. Proteus is not expected, so it's an opportunistic pathogen, so it's a likely contamination. Snodgrassalae is expected, it's a comorbid, it's essential for the biofilm formations and immune modulation. Novocentrophcoccus, no, it's really not well documented in bees. Maybe it's because of the environment. Apilactobacillus, yes, it's formerly lactobacillus. It's commonly associated with bee and bee products. Enterococcus occasionally, and Fructobacillus, yes. So you remember that the genus Lactobacillus have undergone taxonomic revisions, and some species previously classified as Lactobacillus are now placed in Bombylobacillus. And you see now that all the things we do is taking time. So it's really, we do comparisons, manual comparison of the result. And the next question is, what is the taxonomic assignment for this one? So if I look for this one here, can you not find it? BNet 2, this one. I think it was this one. Yeah. And the taxonomic assignations is to Klebsiella. And this one was, if you remember, as a different relative abundance between the two. And so it's Klebsiella, which is really not expected in it might be a contaminations or a clinical, it can be something happening there. And we see that we saw it in the samples that we treated with probiotics. So it could be, the difference could be due to the impact of the probiotic treatments. It could be due to the role of nosema. It could be because of competition there in the ecological niche. It can be because of the functional roles of Klebsiella, so it can be different things. And I'm really not the expert in the honeybee gut microbiota, especially when there is infections. But that is probably something I would investigate more in depth if I would be working on this field. So that's the things we did until now. So we just check that. And the next steps we can do is really adding the relative abundance. Because we see, we just looked at this one. We had to remember where it was there. So we could add the result from cover M there. So for that, we need to do several things. So the first things is we need to replace remove the dot faster here in this column 1. So in column 1, we want to replace the dot faster by nothing. I think it's exactly what we want to do. Yeah, yes. So that's the first things. Afterwards, join two data sets. We want to join the result from this one with the result of cover M. If you remember cover M result, let me follow cover M. So we add for each samples, we add the name of the bean, and then we add the relative abundance, so that we want to combine these two by combining when the names of the bean is the same. So it's what we do, and for that we need to do a join two data sets. We join on the replace text using column 1, and then we give it a collection that is called cover M. No, is it cover M genome? Cover M genome, yes, cover M genomes using column 1. And we want to keep line of the first. Yes, we want to keep line of the first Metagenomics one with the second input. So that's what we want to do. And we can launch that. So that will create a list of tabular data sets, one with width one for each of the samples that will have the bins, the taxonomic levels, and then the relative abundance. But then it will add an extra column. So because when we join, we compare the column 1 and the column 1, and it add again these two columns. So we want to remove the extra column that will be, in this case, after this column here. So that will be, we will have column 1, so 1, 2, 3, so until 8. And then again, the bin number, and then the relative abundance. So we want to remove this column 9. So that's what we do there. So we want to keep column 1 to column 8 and column 10 on the collection join to datasets. That's what we do. Then we will have these 9 columns. Then what we want to do is it will still keep this information here that is a bit strange, user genome classification. So we want to remove the first line. So removing beginning. So we want to remove the first line of the cut on the cut here. Okay. And then what we want to do, yes, now is we have a collection with 2 elements. We have a collections with 2 files inside, one for each samples. And what we want is one file with the taxonomy and the relative abundance for each of these 2 sample. So now from this collection, we will create one table. So we will column join these multiple data sets. So what we give it here, I lost it. Removing beginning, so that will be the same. We give it the collection. We say the identifier column is the first one, and we want to add column names to either. We will manage to it. No, that's we will have now one table with the bin, the different taxonomic level, and the abundance for the different sample, the 2 samples there. So here we have the different sample, and here it's 2 samples. But join 2 times. So, So we have 2 times this information. So we will have column 1 to 9 will be for the first samples, and then column 10 to 17 will be for the second sample. So we want to remove the redundancy. So we will cut and keep only the column 1 to 9 and the column 17. So we need to cut again. So here I copy 1, 9 to 17. And we want to do that on the column join here. And then afterwards, what we want to group, and that we will need to wait until it's finished, because we want to group things, information for the different taxonomic level. We want to group at the level. Yeah, true. I want to know at the genus level, compare the information at genus level. So because you remember, here we have the information at the general level, and we want to compare the abundance for these two at the genus level. So what we will do is we will have the numbers of marks for the first samples, the number of marks for the second sample, and then the abundance of the marks for the first sample and the abundance of marks for the second, for the different genus. And then adding the header, so this header to give the information, and then we will see, so by concatenating, and then we can sort that information. And we should get that information that we can compare then to paper, to the publication in the paper where they looked at the genus level here. Yeah. I will stop the recording while it's still running and come back once it's finished. That is finishly done. So what we have is really a table with the bin names, the different taxonomic level, and then abundance information. And now, as I mentioned, what we want to do is to group things. So group on the genus level, which is column 7, if I'm not mistaken. So here, yeah, the genus 7. And then what we want to do is we want to have the sum count, count to count the number of marks on the column 9. Then we want to count the column on the column 10, so same information. Then we want to do the sum to create the abundance sum that would sum all the abundance on column 9, and then the same sum but on the column 10, and then we can run it. And then we will add this new header to remember what is it. So for that we upload, we create fast fetch, we do that, we call it header, we add that it's a tabular file. I think that's all. And we can start. It should be okay. Like this, if I'm correct. Tabular header. Yes. And then we will concatenate both, concatenate datasets. So the header with the group. And then we can sort afterwards. Sort the concatenates number of either one. We would sort, we need to wait until it's finished. But we will be able to sort that and rename it then. Then when we do that, we can compare what we see in the paper. So in this paper, in these figures, with can we find it? And yeah, I will pause again the recording while it's ready. It's finished. So we now have, you see, the genus, the max numbers for both samples, and the abundance. Now the last steps was to sort, if I remember well. So we want to sort number of read lines, and we want to sort on the column 4. Column 4 by descending order. So to have the most abundant for the first samples on the top. And then what we will be able to do afterwards was to count that here. Rename that. What we can already see if we can see the same genus. So Bartonella is present in the paper, but we cannot find it here, for example. It's not Gracella is there. Fischella is not. Gilliae, we can find it here. Lactobacillus, which has been, no, it's not. Lactobacillus, yes, it is. Then we have Bombilactobacillus, we cannot find here. We have Fructobacillus, that is there. So we can rename that. Rename Chinkans. And here, and we can see that one. So we have Yasnotracella, etc. Bifidobacterium is not found. Enterococcus, we can see there. Klebsiella is not expected, but is found. Novo is found. Proteus, not, but it's listed in the in this panel here. And Serratii, yes. So the core gut is consistent between the paper and the beans we have reconstructed, but some core genera are missing, and some additional genera has been found. The next question is, do the relative abundance of the bacteria correspond to the role reported? So specifically for NP2, so this is the sample 98, and the sample N1, which is this one, N2, so it's hard to read, honestly. But Snodgrassella is the most abundant in both cases, and it's aligned with the dominance in the gut microbiome. The difference is Proteus, which is not a core member of the only gut microbiome and not expected not seen in the paper. Might be a contamination, so environmental exposure, but it's surprising that we couldn't find it then in the paper itself. So maybe misinterpretation here. It's not really similar between the two data sets because missing genera, but so we will need more data to really do a proper comparison. So here was just a quick example to show how GTPTK can really provide a robust and standardized approach to classifying marks and to explore the microbial diversity, compare communities, so using things like I just done, and interpret the ecological roles. So the next steps we can do is really doing functional annotations, which is really a critical steps to identify and characterize the genes that are present in the marks. By assigning functional roles to this gene, we can identify the biological potential, identify metabolic pathways, environmental interaction, host associations, look at the pathogenicity, antibiotic resistance, etc., etc. Found in MAGS, there is different tools like PROCA, BACTA. BACTA is really fast and comprehensive tools for the annotations and of bacterial and archaeal genomes. It can identify coding sequences, so CDS, so coding sequences, ribosomal RNA, transfer RNA, and a lot of other different functional elements. So by default, in the workflow, Bacta was configured to only check for RNA genes to make it really quick to run. But it wants the full functional potential. So for that, I recommend to rerun really the full annotation with Bacta. So let me show you the first result that we had with Bacta. Oh, I can show with that with the multi-QC report. Multi-QC, where are you? I think it's this one. If you look at the Bacta, you see only RNA, which is not expected to have only RNA there. And I can tell you that there is more than RNA in our samples. So we need to rerun the Bacta. For that, we need to give it a collection. We could just relaunch Bacta. That's a good example. So we run, we can do that, we do that, we just say it. We run to rerun, and it will use the same element. So here, I want you to run on the dereplicated genomes. There is an error here. It's not the context file, it's on the dereplicated genomes. The Bacta latest here, MR finder plus latest, output files. We deselect everything, and outputs, we want annotation as TSV, GFF3, raster, summary in text, plots, yes. And we will launch it. And then we can launch multi-QC. After that, multi-QC, because I think it generated a nice report of that. I like the report of multi-QC. Back down, collection, summary text. I think it's this one. Yeah. And then we can launch it, run. And then we can see there. And yeah, backtrack can take one or yeah, it can take sometimes. At least 1 or 2 hours to run. So I will stop the recording and record that later. Bacta has finished now, and MultiQC has run. So we can inspect the result of MultiQC, and we see now that we have assignation for CDS, rRNA, tRNA, tmRNA, and cRNA, etc., etc. So we see we have a lot of features. We have much more information than in the previous run, where Bacta run and this identify only the ribosomal RNAs. So the question we have is, how do the number of coding, total base, and CDS in the provided table reflect the quality and potential functional of the max? So let's look here, percentage. So if we have good quality, it's somehow what is written there. So the number of contigs reflect the fragmentation levels of the assembled genomes. So the more contig we have, may indicate more fragmented assembly, which can complete, for example, as analysis like functional annotation, so comparative genomics. The total number of bases represent the estimated genome size of the max. The larger genomes may encode more genes and have a more functional diversity, while smaller genomes may represent simpler or, I don't like the word simpler there, or more specialized microbes. And they can be used to assess the completeness when compared to expected genome size. And the number of coding sequences indicate the functional potential of the MAG. And so the higher number of CDS suggests greater metabolic and functional capacity. Again, lower number of CDS may indicate a more specialized or streamlined genomes. So this information, the number of contigs, the total base and CDS really provide really important information about the quality and functional potential of the marks. That is important to take into account. So the marks, the 98, sorry, being 2, and the 98 being 3 have the larger number of bases and CDS, suggesting they are more complete and functional diverse. Which species do these beans correspond to, and if the number of predicted CDS consistent with expectation for this species? So we see 90. So it's this one, these two there. If we look, it's this number, they are the biggest. Now we need to go back to the previous, let's get that way here. The previous multi-QC, we will do enable. So we have chicken for multi-QC. Is it this one where we have? No, it's only chicken. So that's the first, oops, that's the stats. We need the web page. I'm just a bit lost. No more. So that should be the last one. It's the web page I want. You see, we can always make mistake. So yes, it's exactly this one I wanted. GTPTK, the taxonomy. Oh, we can also look at the format we had. Where is it? You see, you can get lost within the sample. But that was with the level. Yeah, but I think we will keep this way. So 90 bin 2, it's Klebsiella oxyticae, and the bin 3 is Serratia whatever. I cannot pronounce it. And so Klebsiella, Serratia, both what they talk about, they both were in the completeness. So they have a good completeness, both of them, a good contamination, a low contamination level, so they should be well reconstructed. And if we look on NCBI and we search for Klebsiella oxyticae, so NCBI, you go to NCBI, you search for Klebsiella oxytica, you see here, and then you have where you get examinators. Oh, I remember. I put the link here. So genome. Sorry. Genome here. I cannot click it there. Good. Anyway, I put a link here. I get lost a bit sometimes. So the assembly, this one is one of the assembly, and we see from RefSec and GenBank here we have the genome size, and with the number of contig, we have annotation information with the number of genes here, and the protein coding gene, so the number of CDS that is found in this reference. And if we look at our data, what we saw is we have here for this in there, what were the size? Here it's the size of 5.4 megabyte. And if we look at the reference genomes, it was 5.9, so we are not that far from there. And then the number of CDS that was found with Bacta, please. Why you don't? Here you put it behind. Good. How should I know that? Here, and we see the number of CDS is 4,951. And if we look here, we are a bit above, if I'm correct. Sorry, if I'm correct here. No, a bit lower. Oh, I get a bit lost. So here is what we were expected and what we predicted. So the differences, and it's the same, you can do the same for Serratia. So the number of CDS predicted for both is consistent with expectations based on the reference genomes. We see slightly different that could be attributed to strain level, genetic diversity, or assembly quality, or the annotation method. So at least these two marks seems to be well assembled and representative of their respective species. Then the other question was for the 16 bin 3 and 98 bin 8 that have a few bases encoding sequences that could suggest incomplete genomes. Can we check the so if we look at, so it's 90 here. This is the number of contacts, the number of bases, so we don't really are not, it's a small one. And if we look at, so it's in 316s, we have a completeness that is 86%, and with a low contamination. So it seems to be still a good mark, this one. And it's the same for the bin 8. Good, not that bad contamination, so we are close to 80%. Contamination level is not that big, and again, it's a small genome here. And we look at the annotation. So this one here. So we talk about the bin 3 here. It's a Fructobacillus fructuosus. And if I search for that, Fructobacillus on here, genome, search in CBI here. No, damn it, taxonomy. If we go there, we see that the genome size is a small one, so 1.4, and we saw 1.2 here. So that is somehow expected that this is a small one. And for 8.8 here with its Apilobacillus apinorum, genome, outweigh and take the reference here. And we see again, it's a small one with a small number of CDS. So these marks are also small, and yeah, they seem to be complete, a good completeness, but they correspond to, they represent small genome bacteria. And yeah, it's the results are not that bad then compared when we look at the result there. So the last question is the backed annotation results for 10 marks show the distribution of different features types, including CDS, ribosomal RNA, transfer RNA, and other genomic element. How can we interpret the relative proportion of these features, and what do they tell us about the quality and functional potential of these marks? The answer is, oh, we don't have an answer for that. Good, interesting. So here we have an explanation of the different characteristics in the So the bar chart from Bacta provide visual representation of the relative abundance of the different features. And the number of CDS, so which is in blue when we look in Bacta here. So this is the blue part, the CDS. Yeah, they represent the coding, the potential proteins, and we can interpret with, as we already mentioned, so higher number of CDS may need a more rich functional repertoire and more diversity in terms of metabolisms, environment interaction associations. The ribosomal RNA are for the I mean, I cannot. Yeah, you see, you have a big of explanation for everything, and you can use that to interpret the result of the annotations. But overall, the quality of marks, for the quality of marks, the presence of RNA, tRNA, tmRNA, or RIC suggest that these marks are relatively complete and of high quality. The dominance of CDS and the presence of regulatory element like the non-coding RNA, the CRISPR, or the small ORF indicate rich functional potentials. And the presence of hypothetical proteins and pseudogenes could highlight opportunities to explore more this and maybe identify new genes or functional new functions. So in summary, Bacta annotation can be used to identify metabolic pathway and determine which biochemical pathway are present in the marks. What you can do also once you have all the results, you can use all the results from Bacta, like annotation, the GFF3 to represent them, you visualize them, you using, for example, GBROS or others. You can look at you can really, here you have FASTA files with all the features, nucleotide sequences. So like the tRNA that you have been identified or everything that is great with the annotation. So you can go further with that. You can do further annotations with that. You can explore that. You can identify more things with that, with it. You can go deeper into the results themselves. You have a summary of the things with what has been identified, which proteins or which CDS would that correspond to what. You can really do more in-depth analysis with Bacta. You can explore virulence factor, you can compare potential functional potentials. So the annotation with Bacta is really a powerful approach to look at the biological roles and functional potential of each mark. So it's really crucial and essential for comparative genomics, biotechnology applications, and understanding microbial contribution to ecosystem health. And that is the last steps of the tutorials. Once you have done that, you can do also further annotations, and you're using following some, for example, the bacterial annotation tutorial that we have. So you can run them on each marks, things you will run on individual bacterial genomes. So you can do the same. We can do also virus detections. We are currently developing a workflow for that. And really with these marks, you can do much more with that. Now you have marks that could represent genomes. You have looked at the quality of them. You can identify and choose which one you want to use or you want to explore. You can do much more information with that. And we hope that with this tutorial and this workflow, you can build your own marks and explore your data, explore your community, etc. And some key points, so really important, keep in mind of preparing your metagenomic reads to include the quality control and host contamination removal to be sure that your downstream analysis are accurate and reliable. Assembly and binding are instruction steps to transform raw reads into marks, first in contigs and then in marks. So high quality marks are essential for accurate analysis. The completeness and contaminations are key indicator of the marks quality. When you have high quality marks, you can have more reliable taxonomic assignment and functional annotations. But low quality marks that could be also could be useful also to explore, maybe because it's a completely new genomes that are not in the taxonomy yet. So keep in mind of keeping an eye on the low quality marks. Taxonomy and functional annotation can reveal the biological roles of the microbes by identifying the lineage and especially the known lineage. That's the most important things. So it's based on existing database or based on our knowledge about the taxonomy right now. Same for the annotation, so that helps a lot to identify the metabolics and potential metabolic and ecological roles of your community, but it's also biased by the knowledge that we have at the time we do the analysis. So this globally, it's like metagenomic analysis are really iterative processes. It involves multiple interconnected steps to process, starting from your reads to get taxonomy of functional annotations. Uh, it's important to understand what each step is doing, to understand its bias and be able to interpret the result. So I really encourage you to go through each of the steps, try to understand more. We gave a lot of information in this tutorial that you can read from, but you can also read from the papers, from other sources. And be sure of looking at the result of each steps before running into conclusions. Yeah, and tools and database evolve. So everything that we recorded now might be outdated in 2 or 3 months already. Not much, hopefully, but the numbers could be slightly different. So don't be afraid by that. It could happen. And for that, thanks for listening for this podcast. I hope you learn useful tricks and things about for building your marks and annotating them. And we hope that it will be useful for you. Thank you and goodbye.